Hello, everyone. Thanks to organizers for letting me give this talk. It's actually a good conference. A great pleasure for me, and thanks for putting me in this place where actually a few talks before me are actually greatly useful in introducing some concepts that I will be using here as well. So this is about our latest adventure in trying to understand what enhancer sequences are and what they aren't, and kind of a little bit about the background, like what led us to these questions. So my background is in mathematics. I was happily studying gene regulatory network models where you have these kind of nice wiring diagrams linking target genes with the regulators and trying to understand the semantics of how those multicellular organisms are arising. And then we sort of tried for quite some time now also integrate these with these enhancer sequences and kind of slightly differently maybe from what the previous speaker was talking about. To me, the fascinating feature of enhancers is that you can actually cut them out of their context, of the wonderfully complex context of chromosomal interactions and CTCF loops, and you can actually test them in a reporter assay, and the cells will recognize whether this piece of DNA, this piece of ACG and Ts, is actually an active enhancer or not. So now our model. So over the years in my research, we've been doing these kind of which sequences will be enhancers, which will not. This is actually very similar in spirit, but just from a very primitive 10 years ago approach to learning enhancers from the very early ChIP-seq data sets. This is actually showing that 10 years ago it was also H3K27 cell was the most important feature in terms of predictive power for enhancer activity. So we sort of tried to use different features. Later on, we've kind of, among other people, tried to also understand in larger genomes how to identify these sequences. We've used kind of various different methods of classification. This is a result where we use random forest classifiers and k-mers as features in addition to histone modifications, also being able to identify them. But recently, we've been involved in a study where we have actually collected large data set from a number of brain tumors surgically extracted from patients, and then these were then analyzed, and we had a number of different ChIP-seq tracks. And also, what was the advantage? We had these patients genotyped, like these samples, not genotyped at the positions of active marks. So we basically knew not only that a given patient has a given H3K27 acetyl peak, but also that the same patient has some variants present in this position. So there is some lessons that we've learned in this process also about the variability and that the promoters seem to be much more conserved in their activity than the promoters between the individuals. But then we try to, so this is the main thing that I want to talk about is our approach to, well, this is actually not our approach. This is a previous this is a paper from, I think, some more than 5 years ago, one of the early applications of CNNs to predicting open chromatin. So we adapted this approach to our data to try and now identify from the DNA sequence, from these, in this particular case, these are 2 kb of DNA sequence around the peak. We put it through a convolutional network to try and see whether it can predict that this particular one is going to be an active promoter or active enhancer. Well, but by enhancer, which is actually a non-promoter active region, or inactive one. And we've created several data sets starting from a relatively small, relatively high confidence set where we had actually agreement between patients' data, including some of the high confidence single or multiple situations where multiple patients had the same variant leading to the same change in activity, and then increasing this to a larger data set where we actually included more and more variants in the sequences with more variants. These are lower confidence because then you have fewer examples indicating that this might be actually, because there will be a lot of random events as well. So kind of maybe just quickly, the networks seem to learn, but maybe not all the classes at the time. We needed to modify the architecture, just kind of slightly increasing the size of the fully connected layer. That's, I guess, maybe not that interesting. And kind of, it seems that not only these networks kind of, not surprisingly, were able to learn the kind of patterns that we have been showing them, but also that they were outperforming these more classical methods that we've used before for the task. So now, the most interesting message, the kind of things that we've learned from this for us were actually what are the differences between these networks, kind of going from those so this custom one is actually the one that is learned from the smaller high confidence data set to this kind of larger. And the main thing was actually that when all of this the training was done on balanced data sets where we matched the number of positive and negative examples. But then when we ran them on the whole chromosomes to see what exactly do they predict, turned out that those networks trained on the smaller sets without the information about the variability were actually quite highly predicting false positives. So they like half of the genome for this network looked like an inactive promoter, and this you can see is still prevalent in those in the networks learned on larger data sets, but it's much better. I mean, we've been looking at some examples where we can see some cases where we can actually look into the biological consequences of the specific enhancers being turned on and off by different variants. But I guess maybe this is not so interesting for this crowd. But the thing we've looked more into, and I think maybe this is something that was interesting to me and maybe to some of you as well, was that trying to understand what those convolutional filters are actually representing. So what are these networks basing their prediction on? And as many other people before, we've looked into those kind of PWM-like representation of motif-like representation of those filters. We split them into smaller ones and then compared to transcription factors, and many of them do much. And there's some information in that. But importantly, when you look at this plot here, so this actually, there's three parts to this. So the black part is those parts of the filters that are actually matching some transcription factor. The white part is those k-mers in our filters that are really low on information content. So places like here where you actually don't think that the network is really using this part to drive the prediction. And then there's this gray area, which is actually twice as large as the transcription factor part, which is there is some information, but it does not seem to be important. I mean, it does not seem to look like any transcription factor motif in the database that we have. So we've thought about interpreting this, and actually what it turns out that in all of those networks, the same pattern emerges, is that you can actually identify for those k-mers, you can actually identify high correlation for all of them with various parameters of those DNA shape descriptors. So for those of you who are not familiar, this is research by Remo Ross and other people into measurements of actually what are the preferred shapes and preferred kind of geometrical parameters of short k-mers based on the nucleotides that are there. And I think that in this case, it is actually an interesting thing that maybe the way those networks are learning about what they are learning may be corresponding to some of those lower level. So these are kind of different kind of spacers. So you'd have some binding sites for transcription factors. You had some spacer just saying, well, there doesn't need to be anything there specific on, or you could have just some place where there needs to be a bendable piece, or there need to be a piece with a minor groove of a certain width. So the last thing I wanted to show is actually that we didn't really find, because we wanted to see maybe the major features or things that are shared between the networks, and it seems that there's maybe we've given those models too much flexibility in the model selection. But basically, when you just try to cluster those filters between the networks, we get the best I've ever come in my research to the representation of white noise. So we try to see if this is just overfitting and kind of we just have very different models. But turns out that those networks, despite the fact that they're so diverse in the shape of their filters, they actually agree in majority of cases when they disagree with the label in our training data. And moreover, when they disagree, when most of these networks disagree with the labeling in our training data, they seem to be right. So we've based the activity labels in our training data on the H3K27 acetylation. But when we look at H3K4 trimethyl, which is the well-known promoter mark, we can see that when the network is predicting mislabeling from active promoter to non-promoter active, you can see that those sequences in purple are actually matching the control distribution of H3K4 trimethyl for non-promoters rather than promoters, and vice versa. In the case where the network is predicting these are promoters, the purple distribution is also matching better with the promoter distribution. So to summarize, we think that this was actually a useful exercise, and obviously not surprisingly, we can use these novel neural networks to make predictive models. We're still not there in terms of fully understanding what exactly are the features that are important for making a sequence an enhancer or not, but definitely we will be looking closer at the DNA shape features and trying to give more attention to interpreting them as the key ingredients of regulatory sequences. Thank you for your attention. Yes, I should mention this is mostly work by Magdalena Osipowicz and Magda Machnicka from my lab. Thanks.
Thank you, that was really interesting. And my naive question is, when I hear your very convincing work with the DNA shape, right after the previous talk, there was also a very convincing work about sequence as a clutter. I'm curious what your thoughts are, if both can be true, or perhaps I'm missing something.
Now, thank you for the question. It's naturally so this is from our work. This is 2012. So clearly, you can make these predictions from epigenetic marks, and H3K27 is the most predictive one. So if you do have it, you can predict where in your tissue or cell type. If you just want to know where the enhancers are and you have H3K27 acetyl, you're absolutely better off just using that, or maybe some kind of predictor to kind of remove the noise from your data. But if you want to understand why exactly did I see this H3K27 acetyl peak, so then this does not help you that much. You need to really go down, and especially if you do the exercise, you take the sequence, you clone it, and you see, well, it is active as an enhancer in my reporter assay in the cell type that I'm interested in. So then these models that are based on those histone marks are not really that helpful, right? And it's also true that sometimes the sequence alone will not So if you just train your model in the sequence like we did in the kind of, it will not help you kind of, this will be a very tricky thing. I mean, as we've seen in the first talk, I think from this session, if you have ODN, if you have chromatin openness data, you can kind of, through some clever kind of transfer learning, you can actually make those models. But then what we were trying to do here was kind of go a little bit down into the understanding. So we know that the majority of these sequences could be interpreted by the cell without their context. So if we just clone this, kind of thinking about this in a virtual huge reporter assay. And then the question is, what would be the important thing? We knew about the transcription factors. We knew there might be some positions that are not important, just kind of spacers. And this is kind of like the main conclusion from what we found this time, is that maybe we should take a closer look at these DNA shape, which are kind of very basic, very simple, small features that might actually be the language sort of that is allowing the cells to interpret those